بسم الله الرحمن الرحيم. In this video, we are going to continue our talk about innate lymphoid cells in dermatology. This is another nice paper about the role of innate lymphoid cells in autoimmune disease in the skin. It's published in Nature, March 2020. The title is The Pathogenic Role of Innate Lymphoid Cells in Autoimmune-Related and Inflammatory Skin Diseases. Abnormal innate lymphoid cells functions could contribute to the pathogenesis of autoimmune diseases that affect the skin, example systemic lupus erythematosus and scleroderma. As regards the role of innate lymphoid cells in the inflammatory disease like psoriasis and atopic dermatitis, already I have explained it in the previous video. In systemic LE, we have an imbalance between apoptotic cells and nuclear antigens, extracellular deposition, and between their clearance and disposal. These three nuclear antigens will activate the toll-like receptors of plasmacytoid dendritic cells, which, with the secretion of type 1 interferon, known as interferon signature, and other cytokines, will activate the B cells, which will result to the production of autoantibodies. These autoantibodies will form immune complexes which will attack the tissues and result in a vicious circle and in chronic inflammation and tissue damage. The role of innate lymphoid cells in systemic lupus erythematosus pathogenesis is poorly understood in humans. There is altered distribution of innate lymphoid cells in active lupus, especially with lupus nephritis, which return back to normal after treatments. Some researchers already found an increase in innate lymphoid cells type 1 and decrease in innate lymphoid cell type 3, while others found the opposite, but they were all from blood and not from tissues or organs, which are the main residents of innate lymphoid cells. Here is a simplified diagram about the pathogenesis of systemic lupus erythematosus, where we have apoptotic cells and nuclear antigens that will stimulate plasmacytoid dendritic cells. These plasmacytoid dendritic cells will stimulate T helper 17 pro-inflammatory cells through interleukin 23, and also will stimulate the T cells and B cells. B cells will change to plasma cells, which will then form autoantibodies, and then immune complexes and more tissue destruction and more attraction to neutrophils, and increasing the process of netosis and more apoptotic cells and nuclear antigens, and we have the vicious cycle. Innate lymphoid cells here could have a role through innate lymphoid cells type 1 through production of interferon gamma, which will stimulate or share in stimulation of T cells. As regards the role of innate lymphoid cells in rheumatoid arthritis, in some researches they found that lymphoid tissue inducer cells were decreased in rheumatoid arthritis patients, while innate lymphoid cells type 3 and 1 were increased in rheumatoid arthritis patients in active disease, and in stable disease they found that increase in innate lymphoid cells type 2. Here is another diagram about role of innate lymphoid cells in rheumatoid arthritis, supposed role. So we have innate lymphoid cells type 3 could share in the inflammatory process through production of interleukin 17 and 22, and innate lymphoid cells type 1 could share also through tumor necrosis factor alpha. On the other hand, innate lymphoid cells type 2 could share in tissue repair and in stimulating macrophages to type 2, which is responsible more for remodeling or tissue repair through the production of interleukin 4 and 13. This is a more recent paper in Frontiers Immunology, January 2022, talking about innate lymphoid cells in autoimmune diseases. This is another interesting paper that was published in Frontiers Immunology in June 2022. Its title is tissue-specific diversity of group 2 innate lymphoid cells in the skin. In this paper, they said that innate lymphoid cells type 2 play an important role in maintaining tissue homeostasis, while T cells respond usually to foreign antigens. The activation of innate lymphoid cells is controlled by various tissue-derived factors such as cytokines, lipids, hormones, and neurotransmitters. And actually, innate lymphoid cells type 2 show different phenotypes depending on the tissue in which they are present. The skin innate lymphoid cell subsets in mice show heterogeneity, where skin innate lymphoid cells type 2 that produce interleukin 5 and 13 and express ICOS or CD278 and CCR6 positive but not ST positive play an important role in atopic and allergic inflammation. While the ST positive or ST positive it means interleukin 33 receptor, so ST2 positive innate lymphoid cells 2 are only found in the subcutaneous adipose tissue. This diagram shows us clearly the extent of heterogeneity of innate lymphoid cells type 2 through the different layers of the skin. So innate lymphoid cells type 2 sometimes got characters from innate lymphoid cells type 3, and also they express their transcription factor, retinoic acid receptor gamma T, sometimes through their plasticity and their acquiring characters from each other. Also, innate lymphoid cells, which is ST2 positive, as I said before, is the receptor for interleukin 33, is present only in the subcutaneous layer. So epidermal innate lymphoid cells that express ICOS and CCR6 display characters of both innate lymphoid cells type 2 and 3 and regulate microbiome balance, while inflammatory innate lymphoid cells type 2 that express both GATA3 and retinoic orphan receptor gamma T produce interleukin 5, 13, and 17, which infiltrate the tissues under inflammatory conditions. While interleukin 22, interleukin 17 producing retinoic orphan receptor gamma T and innate lymphoid cells type 3 that expand with ultraviolet induced inflammation also express ICOS and CCR6. The last paper I'm using in this video about innate lymphoid cells in dermatology this paper was published in Frontiers in Cell and Developmental Biology in 2022, and its title Skin Immunosenescence and Type 2 Inflammation, a mini review with an inflammaging perspective. In this paper, they are telling that age-induced immunosenescence predisposes the older age group to pruritic dermatoses, which include type 2 inflammation mediated. Inflammaging means a chronic low level of pro-inflammatory cytokines released from the senescent cells. They said that interleukin 25 and interleukin 33 and thymic stromal lymphoprotein TSLP are released from damaged epithelium, and these cytokines will directly activate the production of interleukin 4, 5, 9, 13, 31 from innate lymphoid cells type 2 and T helper cells type 2, which will lead to type 2 inflammation reaction. This senescence-associated secretory phenotype will lead to immunosenescence with type 2 inflammatory dermatoses like atopic dermatitis, chronic spontaneous urticaria, and bullous pemphigoid. I hope you have enjoyed the video, and again I'm reminding you to subscribe to the channel and activating the alarming bell if you are interested in getting any new video, and thank you.